welcome to everyone, and thank you for giving me the opportunity to present to you today on an area of physiology that's very very near and dear to me, because it's something I've been working on for the last 30 years, and that is basically the endocrinology of pregnancy. How does pregnancy establish? How is pregnancy maintained, and how do women survive pregnancy? How does the female survive pregnancy? And centering on that issue is the placenta and the endocrine factors that are produced by the placenta that alter maternal physiology in order to sustain a pregnancy. And so I'm going to speak today about placental endocrinology, and we'll see where that leads. First of all, I want to say Dr. Bedaiwy mentioned that I wrote the chapter in Yen and Jaffe on fetal neuroendocrine development and the endocrinology of pregnancy. In fact, I inherited that chapter from another famous reproductive endocrinologist, Sam Yen, of Yen and Jaffe. And so I can't claim ownership of that chapter. It's something that I update every couple of years, but it really, the founding, the skeleton of that chapter, the foundation of that chapter was written by Dr. Yen, and I'm very grateful to have the privilege of continuing to add and update that chapter. Okay, so I have nothing to disclose, and let's get on with it. So the endocrine placenta. I want to start by talking about how the placenta develops and how it's situated in human pregnancy, because I think that's very important when you. I'm a PhD. I'm a basic scientist. I did my training using the sheep as a model to study fetal development and the control of pregnancy and parturition. And one of the things that occurred to me very early on in my scientific life was that the placenta is a very strange organ. It is very diverse in its anatomy and function between species. Even though we're all viviparous species, we give birth to live young and we have a pregnancy, we have different placentas, and the placenta can be extremely different between the sheep, the horse, the dog, and the human, yet they serve the same function. And so we really have to put the human placenta into context and realize that the human placenta evolved with hominid evolution, and it's perfectly suited structurally and functionally to deal with the unique aspects of human pregnancy and human development, and controlling the success of reproduction. And so the placenta develops at around the 14th day after fertilization, and it's through the placenta, you all know this, that the baby receives oxygen and glucose and eliminates toxic substances. The human placenta produces multiple hormones that it deposits mainly into the maternal circulation. And I'm going to give you a list of all these hormones in a minute. We're going to talk about some. Obviously, we can't, an hour isn't long enough to go through all the hormones that the placenta produces. Placental hormones modulate maternal physiology, and this is the take-home message. If you're going to walk away from my talk today, really what I want you to walk away with is that the placenta produces hormones that modulate maternal physiology in favor of establishing and maintaining pregnancy and supplying the fetus with nutrients and oxygen. So basically, the placenta is the hormonal control center of human pregnancy. Okay, so placentation. It starts with the blastocyst interacting with the endometrium, and the blastocyst is the starting point for placentation. And the placenta develops from the trophoblast cells, the trophectoderm, that line the outside of the blastocyst. And at no time through human pregnancy does the inner cell mass that will eventually give rise to the fetus, at no time do any of these inner cell mass cells interact directly with the mother. Okay, so the trophectoderm, the trophoblast cells, are the cells that form the fetal side of the maternal fetal interface. So these are very special cells that differentiate very early in development. The blastocyst will travel to the uterine cavity and float freely for a couple of days, and then undergo a process of adhesion, implantation, and invasion, whereby it adheres to the epithelial layer of the endometrium. And the blastocyst cells that are interacting with those epithelial cells will begin to proliferate and form an invasive structure that will penetrate into the stroma of the uterine wall until the blastocyst is completely embedded in the uterine wall. And the trophoblast cells begin to differentiate into two types of cells: cytotrophoblast cells, these purple ones, and this very remarkable cell called the syncytia trophoblast, where these cytotrophoblasts fuse to form one giant multinucleated cell. So this yellow or orangey structure on the outside of the blastocyst is actually a single cell with multiple nuclei. Okay, so we have the blastocyst embedded into the uterine wall, almost as deep as the myometrium, definitely through the endometrium. And it begins to interact with the maternal cells. And again, it's these trophoblast cells on the outside of the embryo that interact directly with the mother. Okay, some of these trophoblast cells form columns that anchor the conceptus in the uterine wall. Others will fuse to form this syncytia trophoblast, this yellow cell. And within the syncytia trophoblast, these vacuoles formed called lacunae. Now, some of these trophoblast cells, the invasive endovascular cytotrophoblasts, will invade into the maternal arterioles in the uterine wall and invade into those spiral arteries and remodel the spiral arteries such that it becomes a low resistance, high volume vessel, allowing an increased amount of blood supply to reach the conceptus. The blood that is coming into that tissue, hold on a second, then fills the lacunae and bathes the syncytia trophoblast in maternal blood. So basically, the maternal blood, the process of placentation involves taking over the arterial supply to the conceptus and allowing maternal blood to directly interact with the syncytia trophoblast and trophoblast cells. Okay, so if we look at this from the point of view of the embryo, we have two interfaces that develop in the process of placentation: the development of the placental disc, and these trophoblast cells that develop the placental disc extend around the conceptus and form what's called the chorion. And the chorion is essentially an extension of the placenta. These are cytotrophoblasts, or they're also called cytotrophoblast cells, that form the chorion layer of the fetal membranes. Now, we could think of the endocrine placenta as only the disc, the placenta we see when delivery happens, but really the placental cells are extending around the fetal membranes, and these cells are producing hormones. They're producing steroid hormones, some peptide hormones. So the placenta, when you think about the placenta, it's not just the disc, it's also the chorion that is surrounding the embryo as part of the fetal membranes. Now, I think this is important because it raises the issue of paracrine, the paracrine placenta rather than the endocrine placenta. And so think of, don't just limit yourselves to thinking about the placenta as an endocrine organ. There are hormones that are produced by the chorion that can act locally to influence the tissues that are right next to the conceptus at this maternal fetal interface. Okay, and we think this becomes important late in pregnancy when pregnancy has to end by the process of parturition. We have a fetal cells, these trophoblast cells, that are capable of producing factors that could affect the myometrium and the decidua, the endometrium of pregnancy, that could lead to the onset of labor. Okay, so the paracrine control of labor through the chorion and the endocrine control of pregnancy through the placenta proper. So if we think of the placental disc, it's arranged in villi, which are these finger-like projections that contain fetal venous and arterial systems in order to carry fetal blood to the placental villi. And these placental villi are dangling or soaking in a lake of maternal blood. Okay, so, and surrounding the placental villi is the syncytia trophoblast. So this is termed a hemochoral placenta because, and as I said earlier, the placenta is very different between different species. And this is just an example of 3 types of placenta. If we look at the primate, the human placenta here, it's referred to a hemochoral placenta because maternal blood is directly in contact with the syncytia trophoblast. Okay, so it's hemochoral, so blood directly in contact with the chorion cell, in this case the syncytia trophoblast cell of the placenta. In rodents, they have an endotheliocorial placenta, where blood from the mother first interacts with the endothelial cells of the mother that then interact with the trophoblast cells. And then, for instance, in sheep, it's an epitheliocorial placenta where blood from the mother interacts with the endothelial cells and then with epithelial cells of the mother before factors can get through to the placenta. Now, I say this because if you think about the endocrine placenta, what better anatomy than the hemochoral placenta for these cells, the syncytia trophoblast cell and the chorion and the cytotrophoblast cells, to secrete factors into the maternal circulation. The human placenta has free access to maternal blood, and so it can put factors directly into the maternal circulation. In contrast, hormones that are made by the mother have to pass through the syncytia trophoblast and the cytotrophoblast and various other cell types before those hormones can access the fetal compartment. And so in terms of the endocrinology of pregnancy, the system is weighted towards the fetus via the placenta, modulating maternal physiology via this direct route to the maternal blood. Okay, so what does the placenta do? So as I said earlier, placental hormones control maternal physiology. They ensure that the fetus is provided with the resources needed for growth and development. And this makes sense. Evolution favors the pregnancy. Reproduction has to happen in a viviparous species via a healthy pregnancy. And so it makes sense that we evolved a placenta that goes out of its way to make sure pregnancy is a success. Now, what are these maternal adaptations? Uterine growth and relaxation is one of them. So the uterus has to be maintained in a quiescent and relaxed state, and it has to be able to accommodate this growing conceptus. Okay, insulin resistance, especially late in gestation. The mother becomes more insulin resistant, so glucose stays higher for a little longer, allowing the fetus to access more of that maternal nutrient. Immune tolerance. It's a very important concept that is only now beginning to get some traction in the research world, because we're beginning to understand how the maternal immune system is controlled such that it doesn't form a rejection response to the conceptus, which is foreign. Okay, and this is mediated by progesterone, estrogen play a role that are coming from the placenta, play a role in modulating maternal immune function. And obviously, I've spoken about increased blood volume and red blood cell and cardiac output going to the placenta. This is promoted by estrogens. Decreased arterial blood pressure happens because the spiral arterioles are dilated, and progesterone has some relaxatory action, so you end up with more blood going to the uterus. And then we have to prepare the mother for the neonate. So lactation is critical for pregnancy to succeed. So during pregnancy, hormones from the placenta promote breast enlargement and the development of breasts that are capable of lactating. And there's some weight gain that happens in the mom, and obviously we need more energy reserves to sustain pregnancy and supporting a neonate. So what are these hormones? As I said, there are many, and in fact it's always been remarkable to me how you name it, every hormone that has been discovered has been discovered also in the placenta. So this is just a list of the hormones that have been detected to be produced by the placenta and mainly secreted into the maternal circulation. So you could pick your favorite hormone and the placenta will be a place to study it. We're going to talk about some of these hormones for the next half an hour. And I've put red boxes around some of them. The steroid hormones that are involved in reproduction, lactation, and to a certain extent behavior. The growth factors, the insulin-like growth factors, vascular endothelial growth factor, and various others that are involved in growth and hemodynamics. The adipokinins are very interesting. I'm not going to talk about them specifically today, but they are involved in maternal energy metabolism and homeostasis, as is placental growth hormone, placental lactogen, and prolactin. And then one of the neuropeptides, CRH, we'll talk about that. But I've selected some of them, but the others are also, as I said, are also produced by the placenta. So I'm not trying to discount the importance of any of these other factors. It's just that we don't have time to cover them all. Bottom line, though, is that during pregnancy, the placenta is producing many, many hormones that it puts directly into the maternal circulation. And then what that raises is the understanding of the endocrinology of human pregnancy. When you think of things like the neuropeptides, the hypothalamic pituitary peptides that are produced by the placenta, that are counteracting effects that the mother's hypothalamic pituitary, say adrenal axis, are producing, it makes you wonder, well, how is the mother maintaining homeostasis when this placenta is producing all these hormones that are getting in the way of regular homeostasis? But she does, and it exists. The problem is that when these systems are pushed to an extreme, one way or another, then we end up with pathophysiology, and I think the next speaker is going to talk about that. Okay, so let's go into depth about some of these hormones. The classic one, Chorionic Gonadotropin, also referred to as human chorionic gonadotropin. I've always wondered why the human needs to be in front of it. I call it chorionic gonadotropin. Okay, so HCG is a very important hormone. It's the hormone that is responsible for establishing human pregnancy. It's made by the trophoblast cells of the blastocyst, and its primary function, it's one of the first hormones that interacts, placental hormones or trophoblast hormones that interacts with the mother in an endocrine manner. Okay, it's produced by the trophoblast cells. It binds to luteinizing hormone receptors on the corpora lutea cells, where it acts as a supergonadotropin to prolong the longevity and the steroidogenic function of the corpus luteum. Keeps the corpus luteum alive, and it keeps it making progesterone. and progesterone is essential for establishing and maintaining pregnancy. It's detectable around 9 to 11 days after the mid-cycle LH peak. It has other actions, but I think its primary function is to act as the hormone that's produced by the blastocyst that keeps the corpus luteum alive, so that progesterone doesn't decrease, which it would normally do at the end of the menstrual cycle to cause menstruation, and instead it keeps the endometrial lining intact in order for the pregnancy to be maintained. Okay, so Chorionic Gonadotropin is analogous to the other glycoproteins, FSH and LH and TSH. It has a common alpha subunit and a unique beta subunit that's hyperglycosylated, and that's what these little symbols are here. And it's this hyperglycosylation of HCG that gives it its distinct function relative to LH. Even though it binds the same receptor, the glycosylation of HCG causes its half-life in the maternal blood to be increased, and its affinity for the LH receptor to be increased. And so it's a supergonadotropin that maintains the life of the corpus luteum. Now, this is important because the progesterone that's needed to sustain pregnancy is exclusively derived from the corpus luteum for the first 6 to 10 weeks of pregnancy. After that stage, the placenta begins making progesterone. The cytotrophoblast cells and the syncytiotrophoblast cell of the placenta begin making progesterone, and after this point, which is called the luteal placental shift, the corpus luteum is no longer required, even though it probably is producing some progesterone for the entirety of pregnancy. It's not essential for pregnancy. So ovariectomy before this stage of pregnancy will cause the termination of the pregnancy, whereas ovariectomy of the mother after this stage of pregnancy, the pregnancy will be fine because the placenta is making progesterone. So how does the placenta make progesterone? Well, in order to talk about the placental steroids, we need to have some understanding of the steroidogenic pathway, and I don't expect you to, this is a very busy slide, but the reason I put it up is because we need to sort of have some grasp of steroidogenesis in order to understand what's going on in terms of placental steroidogenesis. Now, cholesterol can be converted to all of the biologically relevant steroids that control hemodynamics, sugar, glucocorticoid, and stress, and sex. And it does this through specific enzymes that are some of them are branch point enzymes that will metabolize pregnenolone either down through to the mineralocorticoid pathway, or to the glucocorticoid pathway, or to the sex steroid pathway. So the placenta is an incomplete steroidogenic organ. Okay, the cells of the placenta express the P450 SCC enzyme and the 3-beta-HSD enzyme, so they efficiently extract cholesterol from the maternal circulation, convert it to pregnenolone, and then make progesterone. The human placenta does not express any of the other enzymes that will metabolize progesterone, and so the progesterone is then released by the trophoblast cells. Okay, but the human placenta expresses the 3-beta-HSD enzyme, the 17-beta-HSD enzymes, all the isoforms, and the aromatase enzymes. So the human placental cells can take C19 androgens like DHEA and convert them to estrogens. Okay, so this becomes important when we try and understand the fetoplacental unit, which is the mechanism by which the human placenta produces steroids. So this diagram basically shows how the placenta, which is lying between the mother and the fetus, is producing progesterone to begin with. So it takes cholesterol from the mom, it converts it to pregnenolone, and then through 3-beta-HSD to progesterone. and that progesterone is released into the maternal and the fetal circulation. The human placenta lacks the P450C17 enzyme and the downstream enzymes from progesterone, so it cannot metabolize progesterone. Okay. However, as I said, it can metabolize C19 steroids, and so what happens in the human is that for some reason, humans have a very active fetal adrenal cortex that is driven by the fetal hypothalamic pituitary axis via CRH and ACTH that drives steroidogenesis in the fetal adrenal cortex. Now, the fetal adrenal cortex makes mainly dehydroepiandrosterone sulfate, okay, and that DHEAS is converted by the placental cells through the 3-beta-HSD aromatase and 17-beta-HSD systems to either estrone, estradiol, or estetrol. Okay, sorry, estrone and estradiol. I'm getting a bit confused by my own slide. Okay, now the fetal liver in the human also contains 16-hydroxylase, the 16-hydroxylase enzymes and the 15-hydroxylase enzymes. So it can produce 16-hydroxy-DHEA and 15-hydroxy-DHEA that become aromatized to either estetrol. The 15-hydroxy-DHEA gets converted to estetrol, and the 16-hydroxy-DHEA gets converted to estriol. And this is why the human pregnancy contains high levels of estriol mainly, because the liver produces this 16-hydroxylase enzyme. And the liver, this is produced by the liver only during fetal life, okay? So estriol can be used, maternal levels of estriol can be used as an indicator of the activity of the fetal hypothalamic pituitary adrenal axis. So the estrogen producing capacity of the human placenta is remarkable. It has high levels of aromatase and 17-beta-HSD, and it literally just soaks up any C19 androgens coming from anywhere, maternal or fetal, and converts them to estrogens. Consequently, if you look at the levels of these steroids in human pregnancy, they begin to increase fairly early in the first trimester, and you see large amounts of progesterone, estradiol, estrone, estriol, and estetrol. Okay, and what I've put on this diagram is the levels of progesterone in the non-pregnant state, and notice that this is a log axis on the y-axis. So we see that the level of progesterone in human pregnancy is a 10-fold higher than that in during the luteal phase, and the same applies for the estrogens. And if you look at the affinity of the estrogen and progesterone receptors, it's at about the 1 nanomolar range, which is right down here. So basically during pregnancy, women are marinating, they're saturated in progesterone and estrogens. Now, what are these steroids doing? Well, as I've mentioned, progesterone is essential for the establishment and maintenance of pregnancy. It's regulating the uterus such that it maintains the pregnant state. The myometrium is maintained in a relaxed state, the cervix is closed, and the decidua is maintained in a stable state where it's immunologically quiescent, okay? Estrogen, on the other hand, is increasing blood flow to the uterus. It is causing increase in the size and function of the breasts. It's affecting the prolactin secretion from the pituitary, which also is favoring the development of lactation. And so the two together are important for sustaining pregnancy and preparing the mom to nurture a neonate. Okay, so I won't talk much more about the steroid hormones. There's a lot of regulatory factors that are involved, but let's move on to some of the other hormones that are involved in pregnancy. And let's talk about the placental growth hormone, placental lactogen, and prolactin-like hormones, and the insulin-like growth factors. So one of the amazing things, to me at least, about the human placenta is it makes a unique hormone from a specific gene that encodes placental lactogen. Okay, it's also called chorionic Gonadotropin in some textbooks. I think placental lactogen is now the term that is used, that is commonly used. And the amount of placental lactogen that's produced is remarkable, and it gets into the milligram per day amounts when you think of the amount of energy that the trophoblast cells and the syncytial trophoblast cell puts into producing one peptide. It's quite remarkable. And the levels of placental lactogen increase significantly throughout pregnancy, and it's pretty much related to the mass of the placenta. So if you look at placental lactogen levels divided by placental weight, it remains pretty constant through pregnancy. Okay, so we have this hormone called placental lactogen, which is, again, also flooding the maternal system. So placental lactogen is derived from a group of genes that encode for growth hormone and prolactin and placental lactogen. And these genes encode various factors. As I've mentioned, one of them is GH1 that encodes pituitary growth hormone. That gene is silent in the placenta, whereas there's another growth hormone gene, GH2, which encodes placental growth hormone variant, which is active in the placenta only. This is only switched on in trophoblast cells. And then there's the CHS1 and 2, which encode placental lactogen, and these genes are also only activated in placental cells, in trophoblast cells, and the syncytial trophoblast. So during pregnancy, the placenta is producing growth hormone, placental growth hormone variant, and placental lactogen. And the levels increase exponentially, and they're secreted preferentially, as with other placental hormones, into the maternal circulation. Well, what are they doing? So what we think these hormones are doing is affecting maternal metabolism. So via interactions with the growth hormone receptor and the prolactin receptor, because placental lactogen will interact with both receptors, albeit with lower affinity than prolactin and growth hormone for their respective receptors. The fact that the maternal system is flooded with placental lactogen means that it has the opportunity to tweak or signal through these receptors to cause changes in physiology. And what it's thought that the placental lactogen is doing in the mother is that it's stimulating IGF production and causing peripheral insulin resistance, which causes glucose intolerance and makes more glucose available to the fetus, especially late in gestation when the placental lactogen levels get very high. It's also causing lipolysis, and in the breast causing lactogenesis. And the proteolysis that is caused by the peripheral insulin resistance and the IGF production releases amino acids that could also be used by the fetus. Okay, so it's basically a system where we are extracting resources from the maternal compartment. Okay, and this is just a diagram of the growth hormone levels during pregnancy. You can see placental growth hormone here starts going up in the first trimester, and then by the third trimester it goes up very rapidly and then decreases soon after birth. Whereas pituitary growth hormone in the mom is secreted in a pulsatile manner normally for the first trimester and halfway through the second trimester, but then its secretion starts to decrease. Now, this growth hormone that's produced by the placenta is also interacting with maternal growth hormone receptors, but it's secreted in a continuous manner. So the pulsatile signaling that's happening from the maternal pituitary is lost during pregnancy. And it always amazes me, if you look at the levels of placental growth hormone that are produced during pregnancy in a woman why she doesn't develop acromegaly, but she doesn't. And it's probably because of the continuous production or some other factors that might be regulating growth hormone signaling to avoid the phenotype of acromegaly. So placental growth hormone, let's see, what have I not said here? It's mainly secreted during the third trimester. It decreases progressively from about 15, this is pituitary growth hormone goes down and it's non-pulsatile. Okay, so what's it doing? So the theory is that that it's a food fight between the mother and the baby. So after each meal, the mother and the fetus compete for glucose in the blood. Later in pregnancy, maternal glucose and maternal insulin are high after every meal because of insulin resistance. The longer it takes for the mother to reduce her blood sugar, the bigger the share of blood sugar can be taken up by the fetus. And this is thought to be controlled by placental lactogen and placental growth hormone. And when it goes wrong, it can lead to gestational diabetes. And this is where the endocrinology of pregnancy and the endocrine placenta, when it goes a little off kilter and moves one way or another, we end up with an adverse condition of pregnancy and pathophysiology. Okay, so let's talk about other hormones that are produced by the placenta. One of them in particular is corticotropin releasing hormone and ACTH, the POMC, the pro-opiomelanocortin hormones that are each produced by the placenta. Now, normally, these hormones are produced in the hypothalamus, produces corticotropin releasing hormone that then stimulates the pituitary to produce adrenocorticotropic hormone that then affects the adrenal cortex to make cortisol, that then has a negative feedback effect. Well, in human pregnancy, the placenta produces large amounts of corticotropin releasing hormone, especially during the third trimester. And this is just showing the levels of CRH going up almost exponentially a couple of months before delivery. And it also produces a binding protein that is thought to sequester the circulating CRH and make it inactive. That actually goes down as the placental CRH is going up, which means that the bioavailability of CRH is likely increasing during the third trimester of human pregnancy. The CRH that's produced by the placenta is identical to the mother's hypothalamic CRH, and it's released mainly into the maternal compartment. It can be detected in the mother from about the 7th week of pregnancy, and it increases through gestation, reaching maximum levels at term. Now, one of the interesting aspects of placental CRH production is that usually in the pituitary, in the hypothalamic pituitary axis, as I said, cortisol will inhibit the production of ACTH and CRH. It has a negative feedback effect. In the placenta, however, cortisol stimulates CRH. So now we have an inkling of possibly a positive feedback loop, which I'll get to in a minute regarding how CRH might be involved in the regulation of parturition and the timing of birth. So CRH coming from the placenta, which is indicated by this box here, can act on the fetal pituitary to increase its production of ACTH, which will then increase cortisol production by the fetal adrenal, which can then increase CRH. So here we have a positive feedback loop. But the ACTH can also increase DHEAS production by the fetal adrenal, which can be converted to estrogens by the placenta. And now you've got estrogenic drive that could increase the expression of genes in the uterus that could increase myometrial contractility and trigger the process of labor. Interestingly, many years ago, what we found was that CRH also increases the capacity of the fetal adrenal to make DHEAS. So it's the CRH that's coming from the placenta is promoting estrogen production and also potentially promoting fetal cortisol production. Now, the cortisol is important because the cortisol coming from the fetal adrenal gland is critical for maturing the fetal lungs. And so this is a mechanism potentially by which an endocrine factor produced by the placenta is playing a role in triggering the timing of parturition. So the circuit diagram becomes even more complex if you throw in placental ACTH in this mix, and the fact that the ACTH can also act on the maternal adrenal which will make more cortisol, but the estrogen coming from the placenta increases corticotropin binding globulin corticosteroid binding globulin in the mother, and thus it soaks up a lot of this cortisol. So there's a lot of interacting hormonal axes that are involved in maintaining this endocrine homeostasis of pregnancy. But the one thing I want to point out from this slide is that the cortisol that is produced by the maternal hypothalamic pituitary adrenal axis, even though it's influenced by CRH, that cortisol rarely accesses the maternal circulation. Okay, so because the placenta forms a barrier to maternal cortisol. If you look at the list of hormones, I won't go back to that slide, but the list of hormones that's missing on the list of steroid hormones is glucocorticoid, cortisol. The placenta does not make cortisol. However, it metabolizes maternal cortisol to prevent it from entering the fetal compartment. And we think that's important so that the fetal hypothalamic pituitary adrenal axis can develop independently of any signals that may be coming from the mother that is responding to the environmental stresses that she has to live in. Okay, that fetal HPA axis has to be ready to go as soon as the baby is born. And so it can't be influenced too much by what's happening in the maternal environment. It has to develop independently to a certain extent. Okay, so that's why I've put in here this cortisol to cortisone and the 11-beta hydroxysteroid dehydrogenase 2, which is produced by the syncytia trophoblast and the trophoblast cells that protects the fetus from any maternal cortisol. So how does this relate to the hypothalamic pituitary adrenal axis is a stress axis, way that the mother responds to stress. So one hypothesis is that under low stress or normal stress conditions, the HPA axis is producing cortisol that is acting on the placenta a little bit to stimulate CRH. It's not getting through to the maternal compartment, and that CRH is increasing in a trajectory that's related to the mass of the placenta, just like placental lactogen. And eventually it will trigger the cortisol production by the fetal adrenal gland. And under normal circumstances, this pathway becomes activated at term. If it's hyperactive in cases of maternal stress, it's possible that the maternal HPA axis through the cortisol on the mother's side accelerates the trajectory of placental CRH production. Remember, in the placenta, cortisol stimulates CRH instead of having a negative feedback effect the way it does in the hypothalamus. And this accelerated trajectory of placental CRH could increase the risk for preterm birth. And in fact, if you look at the levels of CRH in the maternal circulation through gestation, if the trajectory of increase is accelerated, that correlates with preterm birth. If it's decelerated, if it's slowed, that correlates with post-term birth. And if it's normal, then we have normal term birth. So what we think is happening is that the placental CRH is integrating these stress signals from the fetus, possibly, and from the mother to produce a positive feedback loop within the placenta and with the fetal adrenal cortex. Okay, that makes cortisol that matures the fetal lungs, and the estrogen that's produced changes simultaneously changes the phenotype of the uterus such that it's producing the hormones or expressing the genes that will promote the process of labor. All right, let me end by talking about another group of factors, growth factors, vascular endothelial growth factors, and placental growth factor, and the process of hemostasis during pregnancy. Now, preeclampsia is an important condition that happens in human pregnancy. It's unique to humans, pretty much, and it's thought to be related to the human placenta and the way that those trophoblast cells are invading into and establishing the placenta early in pregnancy. So it's a combination of pregnancy-induced hypertension and proteinuria, most common and dangerous pregnancy complications. It's one of the most dangerous pregnancy complications. It occurs in about 10% of pregnancies. It's primarily a disease of the first pregnancies. There's no cure aside from delivery. And it's thought to be caused by a shallow invasion of the trophoblast cells into the spiral arterioles early in implantation. Now, what the placenta produces is a series of factors, vascular endothelial growth factor and placental growth factor, and a solubilized form of the VEGF receptor called solubilized FLT1. Okay, and this solubilized FLT1 circulates in the maternal circulation and actually interacts with and sequesters VEGF, allowing placental growth factor to interact with the receptor on the endothelial cells, and this is thought to lead to have anticoagulant effects and lead to the vasodilatory factors that will relax those vessels in order to increase blood supply to the fetus. But when the fetus doesn't have enough blood supply because of the insufficient invasion of the spiral arterioles, then the placenta starts producing more of the S-FLT, which sequesters more of the placental growth factor and the VEGF, leading to a deficiency of that signaling in the maternal blood vessels that leads to a procoagulant phenotype and a vasoconstriction. Okay, so again, the placenta is controlling the maternal pathophysiology or physiology, and when that goes either side of homeostasis, it can lead to pathophysiology, and in this case, preeclampsia. I won't go through that because we're running out of time. So this is my last slide, and again, the take-home message of this presentation is that the placenta is a potent endocrine organ. It's also a paracrine organ in that it will affect the tissues adjacent to the maternal fetal interface. It secretes many, many factors that affect the maternal adaptations to allow for pregnancy to exist, fetal growth and development, appropriate timing of birth, maternal competence to nurture the neonate, and to survive a pregnancy and have a healthy mother and baby to the extent that the mother is healthy enough to have a second pregnancy. Okay, so future pregnancies are very important for our existence on this planet. This are not quite right, then we have pathophysiology. So to understand the pathophysiology and the adverse pregnancy conditions that occur in human pregnancy, we need to understand the endocrine control of pregnancy, which is derived pretty much from the endocrine placenta. So I'll stop there and entertain any questions if you have any, and that's it. Thank you very much.
Thank you very much, Professor Mesanio, for such an excellent and informative presentation. And anyone interested in the realm of gynecology and obstetrics would be much benefiting from such important basic informations. I myself have benefited much from your contribution tonight. We do have a number of questions. I would like to, start with Can you comment on placental mosaicism and its possible effect on placental function endocrinologically?
Oh, no. I think that's the simplest answer there. I've never really considered it. Most of my time I study the normal placenta, and I think that the speaker after me will maybe touch on that, I hope, because
Okay.
I'm no,
Okay,
no.
another question here. Can human placental lactogen or placental growth hormone be used as a marker to predict gestational diabetes?
That's a good question. And, one of the problems we have with the endocrinology of human pregnancy is that the tremendous variability of the levels of these hormones in the maternal circulation. And people have looked for biomarkers for adverse pregnancy outcomes. I don't know that anybody has ever really done a detailed study of whether placental lactogen and placental growth hormone variant can be used to predict the risk for gestational diabetes. Um, my my sense is that probably not, because of the variability in the levels that exist in just a normal pregnancy. But that's a good question, and something that somebody should look into.
Another question here. People talk about the impact of the embryo itself as it develops through the mother, but others actually say that there is a contribution from the maternal side, and they always talk about the seed and soil principle of embryonic development. Could you elaborate on such point, please? Thank you.
Oh, I think that's tremendously important. Obviously, we know a lot about the seed, the embryo, but what's happening in the endometrium in order for pregnancy to establish and a placenta to develop is the other side of the equation that's as important. And as reproductive endocrinologists, reproductive biologists, we know that the steroid hormones, progesterone in particular, and estradiol, condition the endometrium such that it is in a state that is receptive to implantation. But we use those words, but the actual mechanisms are very complex. There's a lot of autocrine, paracrine signaling that happen within that the tissue micro environment of the implanting blastocyst, and the the cells involved are also now beginning to be identified, and their function. And one cell type that I think has been not studied enough are the resident immune cells within the endometrium that must be told to not attack the invading blastocyst and the pregnancy. So the conceptus throughout pregnancy has to be somewhat protected from the maternal immune system. And the mechanism for that is not clearly understood. And so the soil is, I think, the next big thing that we need to understand. And so I agree. There's crosstalk happening between maternal cells and the trophoblast cells throughout pregnancy. And that crosstalk is happening between cells at that maternal fetal interface that involve many different cell types.
Yeah, that could also be very much relevant to when facing a problem of recurrent implantation failure.
Absolutely. Yes.
Well, another question here. People are talking about there are various CG variants, and 3 have been described as Corianic Gonadotropin variants. Could you better explain the different types of CG and the variants? Which one is more important? In what way are they different?
Yes. Again, I'm not an expert on Chorionic Gonadotropin and the variants. I would imagine that there's been a lot of research done on the non-corpus luteum effects of Chorionic Gonadotropin. It affects the adrenal gland. It affects the fetal thyroid gland. It's thought to be involved in promoting the implantation and placentation process. So I think we have a very shallow understanding of chorionic gonadotropin, other than its critical role in maintaining the life of the corpus luteum and its production of progesterone. There's a lot more to it than that, and these variants are probably involved. I don't know how to answer the question more specifically than that, other than to sort of skim over the top. I'm sorry.
That's not right. This is great. Another question people have been hearing lately about the extracellular vesicles, the exosomes, and their potential role in perhaps in the coming future, if not yet fully elucidated.
Yeah.
How would you best explain the role of exosomes to the ordinary, attendees or someone who is interested in future endocrine research?
The notion that a cell will package various factors, RNA, protein, lipids, into a vesicle, release that vesicle, and then that vesicle will shuttle those factors to another cell where it deposits them into the cell, and where they will have an effect, is another form of endocrinology that we have pretty much ignored until recently. And the placenta, we've known this for a long time. The placenta releases extracellular vesicles into the maternal circulation, but it's only now that we're beginning to isolate those vesicles, look at what's in them, and try and understand how they might be signaling to specific cells in the maternal compartment. So this takes endocrinology to another level, and I think it's really cool because it means that a cell isn't restricted to just making, it can package a series of factors that will affect a target cell in a specific way, and doesn't. So there's a lot of biology that still needs to be understood here in this particular field. What we know is that the placenta releases these extracellular vesicles. What we don't know is, how do these vesicles target specific cells, or do they target specific cells? Is it just random? What are they carrying? Is what they're carrying sufficient to cause a change in the phenotype of the target cell? But I think it's a tantalizing area because it takes endocrinology to the next level, a level that we hadn't realized was existing until these vesicles were identified. It's at the moment, the research is very exciting, and I encourage anybody who's interested in this area to get in on the ground floor, because I think it's going to be big in the next 10 years.
That's great. Talking about oxytocin as a pituitary-like hormone secreted by the placenta, what is its potential role before parturition, that is, during pregnancy?
Yeah. So oxytocin is produced by mainly the maternal posterior pituitary, but it is produced to a certain extent in the placenta. And the question is, what's it doing? Well, for most of pregnancy, the target cell, specifically the myometrial cells of the uterus, aren't expressing much oxytocin receptor, so they're not very sensitive to oxytocin. It's only when parturition is initiated that estrogen signaling in the myometrial cells will drive expression of the oxytocin receptor. So then the oxytocin will have an effect to cause contractions, but it really is mainly the pituitary oxytocin that does that. So we really don't have a good understanding of what placental or chorion oxytocin is doing. It may be affecting the decidual cells. There has been some evidence that it could be acting on the maternal ovary to in a feedback loop, in a sense, to regulate the corpus luteum early in pregnancy. But in terms of the process of parturition, it's mainly the maternal pituitary oxytocin that's involved in regulating the process of labor.
Yeah, another question, sir, is how do placental hormones act in an immunomodulatory fashion to the maternal side to accept this semi-allergenic allograft?
Yeah, that's a great question. And again, that's an area, as I said, is only now beginning to be looked at more closely. So this issue of immune modulation during pregnancy, one hypothesis is that progesterone acts on maternal immune cells to cause a generalized immune suppression during pregnancy. The other hypothesis is that the trophoblast cells themselves express specific HLA molecules that will camouflage themselves from the maternal immune system. I think it's a mixture of both. The placental trophoblast cells are able to implant ectopically. They don't have the embryo can implant in the fallopian tube. It can implant in the gut wall, the omentum. And so ectopic pregnancies are telling us that the trophoblast has an innate immune modulatory and immune protective camouflaging mechanism where it can implant in an organ or a tissue that is not progesterone responsive. Okay, so the point I'm trying to make is that the endocrine control of the maternal immune system in order to establish and sustain pregnancy, and the paracrine control of the maternal immune system, whereby the blastocyst itself controls the local tissue level resident immune cells wherever it's implanting, probably play equivalent, if not important roles, if not equivalent roles, in establishing the pregnancy and maintaining it. But the endocrine side of it, at least the steroid hormone, favors the pregnancy occurring in the uterus. The fact that the pregnancy can occur outside the uterus means that to me at least, tells me that the blastocyst has an innate ability to overcome the maternal immune system and protect itself from being attacked. The mechanism, I have no idea. And I think people are looking at that intensely. That's another space to look out for in the next couple of years.
Well, we still have questions there. What's the role of the chemokine receptor CXCR3 in relation to recurrent miscarriage and preeclampsia?
I have no idea.
Okay, people have been talking about the transcriptome and the secretome. What's as an integral part of how to control the process of placentation and embryonic development? Could you better explain, how would you explain such terms to a beginner in reproductive endocrinology?
Yeah, well, the transcriptome is basically the messenger RNAs that are present in a tissue or a cell. The secretome is all the proteins and factors that are secreted by the cell. These terms get thrown around, and there's an OM for everything now. And I really think we need to be a little bit more circumspect about how we use these terms, especially as we're getting more granular in the way that we're able to do single cell transcriptome now and single cell proteomics. We tend to sort of lump everything in an OM without realizing the complexity that's going on. And so I guess the question is, how do we use all the omics data to integrate those data into a systems approach to understanding the process of embryogenesis, implantation, and the maintenance of pregnancy. Again, I think we're at the beginning of this field. even though it's been around for 10 or 15 years. The biology is just now catching up to the bioinformatics and the ability to extract massive amounts of data. There's been a lag between taking those data and turning them into specific mechanisms. Because, I don't know if any of you out there have ever done any research involving transcriptomics or metabolomics or whatever omics. You end up with massive amounts of data, and each one of those data points is a potential hypothesis that should be tested. So it's going to be a while for the bench biologists to catch up with the amount of data that's generated by the omics world. I don't know if that answers the question, but I hope it sheds a little bit of light on it.
Well, thank you very, very much, Professor Mesanio, for such a very informative and fruitful discussion.